Are you able to see this slide?
Yes.
I think in previous classes, penicillium also completed from vegetate to structure. So anyway, hello? Penicillium started or not? Right. So penicillium fungi is a saprophytic fungi. What is saprophytic? Saprophytic meaning they can feed the material, nutrition from the death and decay particles. They are called saprophytic. And if you see the vegetative structure, the vegetative body of the penicillium called as mycelium. The mycelium branched, we can see in diagram. Straight. The mycelium, this is the completely mycelium structure. So this mycelium made up of different type of hyphae. So this mycelium branched. This is completely mycelium. These branches are called hyphae. These all branches, all hyphae together called as mycelium. And this each cell called as a hyphae, right? And this mycelium septate. The mycelium septate look like for example, this is mycelium hypha, septa present. The septa with simple pore, remember, septa with simple pore. Here also, septa with simple pore. Like this, the entire mycelium made up of. The entire mycelium made up of like this, right? So here given this highly branched and multinucleated and septate. So let's change the color. So within the mycelium, number of nuclei present, number of the blue color for dots are called nuclei, number of nuclei present, and also septa also present. This is septa also present. So this thread-like structures known as called as hyphae. So the branch called as hyphae, altogether formed as a mycelium. The cell wall, after the mycelium, in next we discuss about the cell wall. This is the order, already told you. In general character, in any type study, first we discuss the habitat distribution, how they grow. After that, how they are, the structure of the mycelium, the structure of the vegetative body. The vegetative body made up of the cell. So the next, after the mycelium, after the vegetative structure, we should learn about the cell. In cell component, the first one, the outer layer, that is called as cell wall. The chemical of cell wall, which is made up of glucose, polysaccharide, and chitin. And next to the cell wall, cytoplasm present. And actually, the cell after the cell wall, plasma membrane, after the plasma membrane, cytoplasm present. In cytoplasm, there all cell organelle present inside the cytoplasm. All cell organelle present. What are the cell organelles? Previous class also we discussed. What are the cell organelles in cytoplasm? Hello. Cell organelle. Mitochondria, Golgi complex. Hello. Fungi cell organelles.
Sir, I don't think anyone remember this, sir. Please, you carry on, sir.
How many class did you attend?
Sir, this is my first class, sir.
Just wait.
Sir, this is my first class, sir.
Okay, just wait, just wait.
Okay.
Renuka Ashok, Sony, Sai Priya, please respond. The fungi cell organelle. Sri Lakshmi. I am asking general topic, general question. Cell component in order for cell wall, plasma membrane, in protoplasm, cell organelle present. Golgi complex, lysosome.
Sir, in fungi, plasma membrane, ribosome, nucleus,
Right,
mitochondria, mitochondria.
right. Yes, they are all present and cytosomes, chromosomes also present. So this much, this is about the cell structure. Right, inside the cell, the cytoplasmic continuity maintained through the central pore. Already told you, the pore present in septa. Through septa, the cytoplasmic streaming flow one cell to the another cell. So this is continuously happened. And after mycelium, after vegetative structure, what are formed? That means the mycelium reach maturity, what are produced on the mycelium? In order, what we learn next? First in habitat, after the thallus structure, cell structure completed. Next, when the thallus or mycelium, a vegetative body, reach maturity stage, next we are going to discuss about what should be the next heading. What would be the next heading? Reproduction. Here the mainly reproduction, two type. Main, of course, three, but is the majorly two types: asexual reproduction and sexual reproduction. First, let's complete about asexual reproduction here. So, before asexual reproduction, in mycelium, after vegetative body, on the vegetative thallus, the reproductive structures also formed. The reproductive structures called as conidia. Reproductive structures are, these asexual reproductive structures are called conidia. This conidia produced on conidiophore. This conidiophore present at the branch of the ending along with the spherical conidiospores, which are asexual spores producing exogenously. We will discuss later in asexual reproduction. In mycelium, vegetative body, after the apical part, these specialized branch are emerged. This specialized branch called as conidiophore. This conidiophore end with conidia. These are called conidia. End with conidia. These are conidiophore. The conidiophore also branched. We will discuss about the formation of conidiophore and formation of conidia. For anywhere, remember the conidia formation not endogenous, exogenously formed. Exogenously means outside formed, not inside. Now let's discuss about asexual reproduction. In asexual reproduction takes place by conidia formation. Already told you. And this conidia developed on, hello. This conidia developed on conidia, right. Right. So now, is it clear? Are you able to see this diagram? Hello?
Yes, sir.
Right. So the asexual reproduction takes place by, actually asexual reproduction takes place by in the special structure called conidiophore. But actually remember the asexual reproduction takes place by conidia formation here. Conidium. Now, conidium developed on conidiophore. Remember it. Conidium developed on conidiophore. This conidiophore may be unbranched or branched. If we take the branched one, the branches are, this is the branched, the separate branches, that is called medullae. And the medullae, another specialized branch form, that is called phyllide. Phyllide, conidiophore, two branches, and this phyllide bottle shaped structure called, these are called phyllides. Right. So after formation phylides, the cell divide mitotically, forming conidium. The phylide cells again divide, pushing the first conidium toward the outside. So anyway, after this is phylide, this is first one. After first one, next second one formation just below the first one. Now the second conidia pushed the first one to upright direction. This process repeat, resulting in a chain of conidium found in basipetal succession. This type of arrangement called as basipetal succession. That means basipetal succession, first conidia, second conidia, third conidia, fourth conidia, they are formed. After formation, first one, second pushed up. After formation, second, the third pushed up, second, fourth pushed up, third, fifth pushed up, fourth. Like this, after formation, so these older one top, sorry, right, older one top and younger one base. This arrangement formed. So this is called as basipetal succession. Already the same we discussed in albuco life cycle also, albuco asexual reproduction also. Right, anyway, after formation conidia, the conidia appeared as a chain. We can see here like a chain system. The conidia may be blue or green or yellow in color, or globose or elliptical in shape. When it is exposed out, the conidia get detached from the conidiophore and dispersed by wind. And they get attached and it takes the suitable substratum and sufficient place. After it takes sufficient place, it is germinate. Germination by germ tube formation first, and mitotic division takes place. After that, mycelium developed. Later, it is formed as a branched mycelium. So this is asexual reproduction. Simple asexual reproduction after formation conidium. The conidium again developed into mycelium like this. So this is called as asexual reproduction. Is it clear? Hello? Are you following
Yes,
me? Right. Now let's take sexual reproduction. What is sexual reproduction? What is sexual reproduction?
So the genetic material is
Right. Fusion of gametes. But remember, in penicillium, the gamete fusion not occurs immediately. There are some waiting period happened. The first, before sexual reproduction, what are the sexual organs in penicillium? The male one called as male one. Let me take marker. The male one, this is the male branch. The pink color, this is the female branch. Right? So this the male branch called as antheridium or male gametangium, and female one called as ascogonium or female gametangium. Remember, they are grown side by side like this. You can see in slide. Are you able to see the slide? Sexual reproduction of penicillium. Hello?
No, sir.
Is it not visible? Sexual reproduction of penicillium?
No, sir. कागज सर.
Now visible?
Yes, sir. Yes, sir. Yes, sir.
Right. Sexual reproduction, penicillium. So in this, the male one and female one, at the male branch, small, slender, elongated, spiral manner, that is male branch, which is called as antheridium, and club-shaped, elongated club-shaped structure, that is called female, and this female one called as ascogonium. They are grown side by side, right? And what happened before sexual reproduction? One place it is the antheridium contact with ascogonium. Just contact with ascogonium. Before that, actually here the one nucleus present in antheridium, and also here the one nucleus, different, more than one nucleus present in ascogonium. They are the nuclei. But after contact, what happened? Actually, immediately after contact, the antheridium nuclei does not enter into the ascogonium. The first only the cytoplasm enter into the ascogonium. Now the nuclei enter into the antheridium. Sorry, ascogonium. Nuclei enter into the ascogonium. Which nuclei? Antheridium nuclei. Antheridium nucleus. First cytoplasm, next nucleus enter into the ascogonium. But not fuse. When the antheridium nucleus enter into the ascogonium nucleus, immediately division takes place in ascogonium. As a result, it is the one nuclei become two nuclei. One nuclei form two nuclei. Later formation septa, formation septa between two nuclei. Between two nuclei. Right? After that, from two nuclei part, the one of the, actually not given here, one of the specialized structure emerged. The specialized structure called as crozier. Are you able to see this diagram? Marked diagram. Let me change color. Are you able to see the blue line? Hello?
Yes, sir.
Yes, sir.
Right. So anyway, from the dinucleate part, this is two nuclei, dinucleate. Actually, after antheridium nuclei enter into the ascogonium, there is no fusion. Only the two nuclei formed, two nuclei formed. After formation, two nuclei from the cell, one of the cell. This is called one of the cell. This is one cell. This is another cell. 1, 2, 3, like this. Different type of cell are there. From any one two nuclei or more than two nuclei, more than two septa, one specialized branch developed. This is called ascogenous. This is called ascogenous filament. Ascogenous. Anyway, after formation ascogenous, these two nuclei enter into the ascogenous. Later, this is underwent to mitotic division. After mitotic division, these two become, hello? These two nuclei become, so now
Four.
it is, right, four nuclei formed. So anyway, the four nuclei developed in ascogenous filament, 2, 3, 4. After formation, four nuclei, septa formed. Two septa formed in ascogenous filament, two septa formed, which divide one apical nuclei, one basal nuclei, and middle. As a result, formation of three cell. After formation septa, ascogenous filament formed as a three-celled 1, 2, and 3. The first one, this is called basal cell. This is called apical cell. And middle one, ascus mother cell. Ascus mother cell. Or penultimate cell. First, actually, penultimate cell. Later developed into ascus mother cell. What happened? Ascus mother cell here. These two nuclei fused. Now the two nuclei fused. When the antheridium nuclei enter into the ascogonium, when the antheridium when attached to the ascogonium, then only along with cytoplasm, for cytoplasm after follows the nucleus, but not fused with ascogonium nuclei. They become double, two nuclei formation, two nuclei ascogenous. The specialized branch emerged. The specialized branch called as ascogenous filament. After formation ascogenous filament, the two nuclei enter into the ascogenous filament. Later septa formed. As a result, three-celled form with four nuclei. So later this is apical and basal and penultimate cells are formed. This penultimate cell, only I will take this penultimate cell developed as a ascus mother cell. What happened in this? These two nuclei fused. Only I am drawing middle cell only. Don't get confused, middle cell. What happened in middle cell? As a result, the fusion of two nuclei, it becomes diploid, right? Hello? Now this is called ascus mother cell. Now this is called ascus mother cell. Before fusion, that is called penultimate cell. Before division, called as penultimate cell. After fusion, now this is called ascus mother cell. Right? Next, this ascus mother cell undergoes to meiotic division. Meiotic division. What is meiotic? Hello?
Only one nucleotide is formed after the. Yes,
How many? That means after meiotic division, the ploidy of nucleus.
Haploid.
After meiotic division, ploid, right? Haploid. So now two and diploid one becomes haploid. Becomes haploid. Like this, four haploid nuclei form. Four haploid nuclei form. Later, this four haploid nuclei undergoes to mitotic division. After mitotic division, how many? How many nuclei form? Hello? After mitotic division, four nuclei. These four nuclei enter into the mitotic division. Now tell me the number of nuclei.
8.
8. Now 8 nuclei form in this. So actually this is the part of the penultimate. These all changes occur inside the penultimate. Later the rest of the filament. Rest of the filament surrounded the ascus. Rest of the filament surrounded ascus and developed into fruiting body. The fruiting body called as closed fruiting body clistothecium. You can see in your book page number 162. Turn on page number 162. Turn on page number 162. The figure number 11.2.2. Are you with the book? Hello? Hello?
Yes, sir.
Right, in figure number 11.2.2, see the G diagram. In life cycle given clearly, life cycle A, E, F and G, H. If you see the G diagram like this, the round shaped structure. Within the round shaped structure, the 8 ascospores are there. So these 8 spores called as ascospore, as it name ascomycotina. These spores called as ascospore. So anyway, after formation, this ascospore inside the Cleistocesium fruiting body, when this ascospore maturity reach maturity stage, the wall of the Cleistocesium disintegrate. Wall of the Cleistocesium disintegrate, destroyed. As a result, ascospores released out. Now this applied ascospore germinate into mycelium filament. Germinate into mycelium. And again mycelia form. So this is the sexual reproduction in penicillium. Is it clear? Hello?
Yes, sir.
Hello? Right. So this is also very important question in exam point of view. And next one, another important one, Puccinia life cycle. The Puccinia life cycle you must have learnt in practical training classes. Didn't you? The practical training classes.
Yes,
You
sir.
learn about Puccinia. And also we have taken the section cutting also about Puccinia, groundnut, Ticca disease, albugo also. So
Yes, sir.
again, once again, recollect one, this Puccinia life cycle, because this is also very important. And let's see Puccinia life cycle. Do you know iridinospore, teleospore? Already these words you must have listened. You must have learnt in during practical training classes. So anyway, are you able to see the slide?
Yes, sir.
Right, Puccinia. Puccinia. Are you able to see the Puccinia slide?
Yes, sir.
Let me take a marker. So Puccinia is a very large genus with 8 and 1800 species distributed in all parts of the world, and more than 147 species. The species of Puccinia are obligate parasite. What is obligate parasite? Already told you in fungi nutrition. What is obligate parasite? Obligate parasite means they are grow and always parasite. There are two type of nutrition type, obligate and facultative. Facultative means dual. Obligate means only one side, one type of nutrition. Obligate parasite means actually they are only parasite. If there is no host, they cannot survive. Puccinia cannot survive without the host. And at the other end, facultative parasite. Actually, facultative parasite, not a parasite. Facultative parasite as a saprophyte. When the death and decay not available, at the same time, the living host available, they can turns into parasite. That's it, facultative. Right? And and the species of Puccinia are autoecious or heteroecious. Remember these two words important. What is autoecious means? Just wait. Autoecious and heteroecious. Autoecious species means complete life cycle single host. Heteroecious means complete life cycle to different host. Yeah, actually, we are discussing about Puccina gramineous TTC. The Puccina gramineous TTC not a autoecious. It is heteroecious host. Heteroecious means it can complete their life cycle when two different hosts. Here the two different host means at the primary host and secondary host. Primary host wheat, secondary host barberry, secondary host barberry, primary host and secondary barberry. So in India, wheat crop is infected by the following three species of Puccinia: Puccinia gramineous TTC, Puccinia recondita, Strychnoformis. They are mostly infected the wheat crops in India. And next, Puccinia gramineous casual organism black rust disease, which cause the stem rust disease in wheat. Already told you obligate parasite. It is a heteroecious fungus in wheat. That means gramineous TTC. Already told you heteroecious means two different host. One dikaryotic phase and haplophase. I will explain later these two. So the dikaryotic phase occurs in primary host. Already told you. Primary host is the wheat plant, Triticum eastworm, primary host. And whereas haplophase in alternate host, that is called Burberry vulgaris. So dikaryotic phase means di means two, karya means nuclei, two nuclei present in primary host. Haplo means one nucleus present in alternate Burberry host. That is the different. Dikaryophase and haplophase. And gramineous can survive in the absence of alternate host, but its life cycle completed only when the both hosts are available. This is important. As it obligate parasite, which need even one single host, right? If only one host present, it can survive, right? So remember primary host and alternate or secondary host. Next one. The physiological specialization, the gramineous, Puccinia gramineous has several physiological races which show physiological specificity toward the host. And Puccinia cause rust disease in several cereal crops like wheat, barley, oats. And let's see vegetative structure. There are two types of mycelia. Dikaryotic, just now we discussed dikaryotic mycelium in wheat plant. Monokaryotic mycelium in barberry. We will discuss later about this both. And these both mycelia are intercellular, septate, and branched. They are all intercellular, that means between the cell, intercellular space, septate, and branched. There is a simple pore in each septum, which maintain protoplasmic connection between the adjacent cell. Each cell of the dikaryotic mycelium have two nucleus, and monokaryotic mycelium have single nucleus. Already told you. The mycelium takes nourishment from the host cell with help of the spherical haustoria. That is very, very important. So this is the mycelium. Already as it is parasitic, the mycelium grown inside the host intercellular space. Right? And now life cycle. There are 5 types of spores in life cycle. The first iridospore or iridospore. Second teletospore. Third bestiospore. Fourth pycnidiospore. And fifth thesiospore. Remember first the two iridospores and teletospore on primary host triticum. And bestiospore neither farm wheat nor farm barberry. The bestiospore formed in the crop field soil, only soil. Non-Picnio and Isio both are formed on the barberry. Now first let's discuss the primary stages. Iridio stage, iridiospores. The first iridiospores mycelium is produced by ichthyospore and germination on wheat plant. So first remember, so this is the ichthyospore, right? I will explain later how the ichthyospore developed. First, let's start from the ichthyospore. Actually, after formation ichthyospore, this ichthyospore liberated from the barberry plant, and these ichthyospore infect the primary host wheat. After infection by ichthyospore to wheat, it is germinated, germinative formation, mycelium formed in wheat intercellular space. After mycelium develop, which produces iridiospores, which produce iridiospores. Remember, the first iridiospores are formed. Is it clear? Hello? Be careful, don't get confused. Heteroecious life cycle. Hello?
Yes, sir.
Please follow me. If you have any doubt, ask me, because it is just somewhat some difficult question, some complex topic. Heteroacia 2 host to primary, secondary. First, let's discuss about the primary host. In primary host, first iridiospores are developed, later which are developed as a teletospore. From the teletospore, in crop field, basidiospores are formed. Third, this basidiospore now infect the barberry plant. After infection barberry plant, it is developed a pycniospore. And at the same time, ichthyospores are also developed in another side. Apical side pycniospores and below side ichthyospore. Is it clear first? Hello? This summary clear? This brief explanation clear or not? Only if
Yes, sir.
you clear about it, you can understand the rest of the life cycle. Can I go move forward?
Yes, sir.
Right. So in life cycle first, let's start with primary host wheat. In primary host wheat, what are formed? First, iridiospores are formed. And these iridiospores produced by ichthyospores. I already told you how the ichthyospore germinated and produced iridiospore. So the ichthyospores and germination and wheat plant, which produce a binucleate iridiospore, developed a tip of the each hyphae. The spore developed a group, this group known as iridiosauri. With the formation iridospore, disease symptoms appear on the form reddish brown. Reddish brown. The pustules are streaks on the stems and leaf base. Because these iridiospores red in color. That's why after formation idiospore, the symptoms appear in red in color. The symptoms appears mainly late spring season. The host epidermis burst due to the pressure of developing iridiosauri, and the iridiospores get exposed and liberated out. The mature iridiospores are oval shaped, binucleated. So these are the iridiospores. So let's discuss about iridiospores. First of all, let's take here. So first, this is dikaryotic mycelium, which is developed by ichthyospore. Actually, ichthyospore developed on barberry, and after detached from the barberry, this ichthyospore infect the wheat, and this ichthyospore generate germ tube and form the dikaryotic mycelium. This dikaryotic mycelium form the specialized branch just below the epidermis, just below the upper epidermis. The later, just below the upper epidermis, these iridiospores are formed. These are all iridiospores. These are all red in color. This each iridiospore unicell binucleated with stalk. This is stalk. So after matured this iridospore, red color iridospore, the area appear as a red in color, rust in color. While the growing, the pressure falls on epidermis. This is epidermis. Pressure falls on epidermis. Due to the exit pressure, outside pressure, this epidermis ruptured. Now this iridospore exposed out. Now detached from the wheat, and these are again infect another wheat. Again infect another wheat. Now this is called as repeated stage. That means this is repeatedly formed. This is repeatedly formed. That's what is called as a repeated stage. Now, so they are completed. Now, so once the formed iridospore spread the disease rapidly under the favorable condition. That means repeatedly formed. Now teleal stage. Are you able to see the slide teleal stage? Hello?
Yes, sir.
Right. At the end of the wheat season, actually the wheat formation earlier stage, but it is repeatedly formed. The wheat iridospore repeatedly formed. But end of the season, iridospore also start to producing teltospore. Addition to iridospore. Actually, repeatedly the iridospore repeatedly formed iridospore. But end of the season, instead of iridospore, which producing teltospore, the same postule, the same place, the iridospore are ultimately converted into teltospore and produce teltospore exclusively. The teltospore is stalked like iridospore, bicelled. But what about iridospore? Iridospore, how many cell?
Single.
Single, right? But the difference, the teltospore, bicell, of course, both are stalked, but it is bicell, spindle shaped, but iridospore, globose, round shaped, and constricted slightly at the septum. And the wall of the teltospore is thick and smooth. And the same, the iridospore wall also thick but not smooth. The iridospore wall is somewhat spiny. Spines are there. And each iridospore binucleated is provided with germ pore. As the teltospore mature in two nuclei of the cell fuse to form diploid nucleus. That means teltospore like this, stalk, spindle shape. How many cells it is? And also small constriction. This is constriction. Is it how many cells? Here the small sorry. Two cells. In two cells, two nuclei, plus nuclei, minus nuclei, plus nuclei, minus nuclei. This is formed during harvesting period.
Germination.
So, right, harvesting period, the teltospore formed, but this teltospore not in red color. The teltospore in green in color. That's why the symptom appear in green in color. Actually, first in wheat rust disease appear in red rust color. But when the disease reaches severity stage, turns into black color because of formation of teltospores. And the same place. And the teltospore act as the resting spores after fusion. So actually, what happened after harvesting? The teltospore mature at the two nuclei in a cell fused to form a diploid nucleus. After that, inside the cell, plus nucleus, minus nucleus fused to diploid form. Also diploid 2n form. Both 2n form. Now this is called resting spores. Now it is undergoes to dormant period. Is it clear? Teltospore clear? Hello? We can see diagram here. Double celled, small septa. Actually, it is not. It is single cell means after fusion. Before fusion, actually two nuclei. We can see here in this diagram. Two nuclei. After two nuclei fused and underwent to dormant period. Now let's see the basidiospore formation. These are the basidiospore. This basidiospore formed by teltospore. Sorry, here. So this is 2n. After fusion 2n, this teltospore. This also teltospore. What happened from the germ tube? From the germ pore, germ tube form. This is germ tube. Next, meiosis division takes place in diploid nucleus. After meiosis division, haploid nuclei formed. These haploid nuclei enter into the germ tube. As a result, formation of promycelium. This is called promycelium. After the first germ tube, after formation germ tube, after meiosis, the nuclei enter into the germ tube and form a septa. Now forms promycelium. After that, from the septa, just side the septa, the bulbous-like structure formed. Two bulbous-like structure, this nucleus entered. The four nuclei entered into four bulb-like structures. Now this is called as basidiospores. Now the basidiospores are formed. Now this based after formation, basidiospore. Now next, in fact, what? What is next word? What is the next host?
Host.
Hello?
Yes, sir.
What is the next host? Now all the spores are formed in wheat or barberry.
Barbary.
So far we discuss.
Wheat, sir.
Right, wheat. So wheat, iridospore formed and teltospore formed. After harvesting
Yes, sir.
in crop field, basidiospore formed. The basidiospore, the not related to neither wheat nor barberry, remember it. Is that formed in crop field? So anyway, after formation basidiospore, after matured basidiospore, after detached basidiospore, this basidiospore infect which plant? Wheat or barberry?
Barbary leaf.
Right. Next, it infects the alternate or secondmost barberry. Now let's discuss about barberry spores. Fuchsina gramineous barberry plant. So already told you very beginning the barberry only haplophase. There is no binucleate, only single nucleus present. So that is called haplophase. Now let's discuss pycniospores and next ichthyospore. Now this is the both are formed. Let's discuss. Right. So this is barberry leaf internal structure. If see barberry leaf internal structure, barberry leaf internal structure. This is first of all barberry, sorry, basidiospore. After basidiospore, infect the barberry, germ tube form. This germ tube developed into mycelium in barberry internal. Later, these are found pycnidium. From the mycelium, pycnidium are formed. The pycnidium produce a pycniospore. These rounds are called pycniospore. And also it has the receptive sites also there. Through spermatization, the pycniospore receive receptive site. After receive receptive site, just below the epidermis, the another mycelium, the same mycelium extended as a binucleated and form the chain of ichthyospores, isidium. So this ichthyospore formed in complex structure called isidium. So each ichthyospores are binucleated. Because after spermatization, binucleation takes place. That's why it's binucleus form. Actually, initial stage, this pycnio spores also uni. But after spermatization, it becomes binucleated. Now this binucleated spore next infect. How we start the class? From which spore? From which spore class started? Spore. Now end with spore. Is it clear? Hello? Hello?
Yes, sir.
Right. Let's see the life cycle. Develops a mycelium issues for chem. Are you right? It's not covered in single. Are you able to see the life cycle chart?
Yes, sir.
First, actually, it is not completely clearly appear, but anyway. So first, this is start with ECS4. Life cycle start with ECS4. This is the first one. Okay. So now the ECS4 disseminated by rain or wind. Infect what? Which plant? Infect now it is infect wheat. Here remember wheat. Primary host. Infect primary host. Actually, after infection, germination takes place. Okay. Germination and form the mycelium. Right?
Yes, sir.
Second, mycelium form. Where is the mycelium now? Where is the mycelium? Hello? In
Primary
which?
host.
Right, in wheat host. So mycelium formed. After mycelium formation on the mycelium, iridosaurus formed. Iridosporus called binucleate iridospores. These are by disseminated by rain or wind and germination, stomatal penetration. Again, infected the wheat plant. That's why it is called as a repeated. So this is all dikaryo, 2 nuclear dikaryo phase. Right? Is it clear?
Yes,
Okay.
sir.
But end of the crop, end of the same postule, the same place, Tiltosaurus developed. Tiltosaurus. This is the fourth step. They are all in 3 steps, repeatedly formed. That is all 3 steps. So the tiltospores are bicelled to bicelled teliospores. In bicelled teliospores, karyogamy takes place. Karyogamy. Bicelled nuclei, karyogamy takes place. After karyogamy, what happened? Hello?
Yes, sir.
After karyogamy, it underwent to dormant period. Next, in trough dormant period, next from the diploid nucleus, what are developed? Hello? We don't have enough
Yes,
time.
sir.
Please
Which,
respond.
yeah, teleotospores or mature teleospores are produced, sir.
Actually, mature
Germinal.
teliospore itself, karyogamy occurs. From mature teliospore, what are developed?
Um,
See, karyogamy, the mature teliospore, from mature teliospore to next, meiosis. After meiosis, basidium, basidiospores are formed. Basidiospore, remember, fifth basidiospore. Okay. After basidiospore, this basidiospore germinate barberry leaf. Sixth, the sixth one. The basidiospore plus strain, minus strain, different. What is spermagonium? Sir, minus spermagonium plus spermagonium. The both have receptive site. Spermatium, receptive. So here what happened? The plus spermatium received by minus receptive site. Minus received by plus receptive site, vice versa. This is called spermatization. This type of sexual reproduction called as spermatization. What happened after that? Dikaryotic mycelium formed. In dikaryotic mycelium formed the ECL cup, sixth, ECL cup 7, sorry, echiospore formation 7. The final echiospore form, right? Hello?
Yes, sir.
Just wait. Actually, we start the class with echiospores. Again, form the echiospore, right? End with echiospore. That is the life cycle. Is it clear? Hello? Any doubt in this?
No, sir.
Right. So please draw this life cycle. Just wait. So any doubt?
No, sir.
Right. Please draw this life cycle. Please draw this life cycle. Very important. Can I leave the class? Hello? If you have any doubt, ask me. Just remain almost 90% completed. First semester 90% completed. Only remains the 12th unit diseases. Only crop diseases. Only remain crop diseases. As, and these crop disease also completed during practical training classes. Hello? These crop diseases completed during practical training classes, didn't it? Hello? Hello? Are you there? Am I audible? Okay, thank you all and all the best.